So the generation of humoral effector mechanisms now. What does the USMLE want me to know about the generation of humoral effector mechanisms? Well, I need to know that the humoral responses to thymus independent and thymus dependent, the humoral responses to thymus independent and dependent antigens, cell surface molecules that contribute to the production of T and B cell conjugates. I need to know the basic structures and effector functions of the 5 antibody isotypes. I need to know the characteristics and function of the secretory IgA. What does valence, affinity, and avidity mean? What is the role of somatic hypermutation? Oh, here we go. In affinity maturation. How the alternative and classical complement cascades are initiated. The biologic functions and complement components. So we're going to learn a lot of the things that we've been kind of foreshadowing, I suppose, in the previous chapters. Okay. So let's just start with goals of humoral immunity. So humoral immunity is mediated by antibodies synthesized by B lymphocytes and secreted by their fully differentiated N cell, which is the plasma cell. This arm of the immune response is directed toward the defense against extracellular microbes or toxins. And they may culminate in the extracellular degradation of such materials or the enhancement of their destruction via phagocytosis. So that's pretty explanatory. Basically, B lymphocytes will become activated and eventually become a plasma cell, but before they do that, they're going to start producing antibodies, and that will initiate a humoral immune response. And basically, these antibodies will target extracellular microbes or even toxins, actually, and they will cause extracellular degradation of such materials, or they will cause them to get phagocytosed. Okay, so now as mature B lymphocytes leave the bone marrow following successful rearrangement of their membrane immunoglobulin receptor genes, they recirculate throughout the body, attracted especially to follicular areas of the lymph node and the spleen. If an antigen entering these secondary lymphoid organs binds to and then cross-links the idiotypes of these membrane receptors, this provides the first signal for the activation of the B lymphocyte. So it's a bit of a review there, but basically if an antigen binds to these antigen recognition receptors that with a unique idiotype, that will activate, that will be the first signal for the B lymphocyte, just like TCR binds MHC2. Similar case here. So most antigens introduced into the body fall into the category of thymus-dependent antigens. Okay. Response to such molecules requires the direct contact of B cells with T helper cells and their cytokines. After the cross-linking of receptors on the B cell surface with antigen, the material is then endocytosed and processed via endocytic exogenous pathway to generate MHC class II peptide conjugates, which are then inserted in the membrane just as we have seen during phagocytosis and processing in the professional phagocyte lineages. Simultaneously, expression of the co-stimulatory molecules such as B7 is upregulated on the B lymphocyte, making them effective presenters of antigen to the T helper cells in the area. Once a T8 cell recognizes a processed antigenic peptide displayed with MHC class II molecules on the membrane of the B cell, the two cells form a conjugate, and the T helper cell is then activated and induced to become Th2 cells. Why Th2 and not Th1? We will see. Because here, what are we doing? We are B cell surface within antigen. So it's an APC. Okay, I guess that's a thing. B cells will cause the formation of Th2, perhaps. Okay. Th2 cells in conjugates rearrange their Golgi apparatus towards the junction with the B cell, leading to the directional release of cytokines towards the B cells. That's convenient. In addition, expression of a molecule known as CD40L on the surface of the Th2 cell is upregulated. And this molecule interacts with the CD40 on the B cell to provide the second signal for B cell activation. The B cells are encouraged to proliferate after this interaction. The final signal delivered by the Th2 cell is a release of cytokines, which will induce the differentiation of B cells into fully differentiated antibody-secreting cells and memory cells, and induce class switching. So there was a bit of a review in the beginning. The only thing is that most antigens fall into the category of thymus-dependent antigens. So these thymus-dependent, I'm sure there's a picture coming up. Yeah, there is. So I'm going to do my classic here. All right, then. So here we have thymus-dependent antigen, for instance, comes into the body. And basically, they will come into direct contact with a B cell. And that B cell will present it to the Th cells, and they will both produce cytokines. So the MHC molecule, obviously, MHC2. So they're going through the whole previous process, basically. B cell is an antigen-presenting cell, right? And it's producing, once the variable gene has been made and it's just sitting there on the surface, it binds some antigen, and then so after the cross-linking of the receptors on the B cell surface with the antigen, so basically two of them have to join, say two antibodies have to join, then that material is endocytosed again and processed again via the endocytic exogenous pathway, and that will generate an MHC class II peptide conjugate. Yeah, see, this is where it was getting confusing. So this is IgM, IgD, that's what's on the surface. They will bind to a particular antigen. In the meanwhile, you're producing MHC in here. And while that's being made, the invariable chain is covered up, obviously. Well, this is endocytosed into a phagosome and joined to that phagosome endosome, and that endosome joins with a lysosome, making it a phagolysosome, and then at the same time it joins with a vesicle, with MHC2 molecule. That's degraded. Invariable change of MHC2 is degraded, forming, revealing the variable domain or the binding area of MHC2, where broken up protein particles of that antigen are attached, and then that MHC2 is displayed on the surface as such. Now that MHC2 molecule will present whatever it found, that degraded protein molecule, to a TCR receptor on the CD4 T helper cell. This will induce the cell. And then remember in the previous chapter, we learned how the B cell has to produce a B7 molecule. The same thing happens here. The B7 is also being triggered at the same time. The moment this antigen binds is being released, and CD28 will have to anchor it. Another linkage, this is CD40 and CD40L ligand, basically. In the previous one, we had the fusion of CD2 with the integrin LFA3 on the macrophage. So just not much different except for the ligand. Instead of LFA3, you have CD40. Instead of CD2, you have CD40 ligand here. Well, anyway, once this happens, it causes the it causes this conjugation occurs, and as a result, you have basically induced that T cell to choose to become a T helper type 2 cell, basically. And now this T helper 2 cell will conjugate and then rearrange its internal Golgi apparatus where this junction happens, so that it's closest to this B cell. And so whenever the Golgi is making vesicles filled with cytokines of some sort, they will be, when they're exocytosed, they will be in proximity to the B cell. And so that would be the second signal for B cell activation. The B cells are encouraged then to proliferate, and the final signal is delivered by Th2 cell, is the release of cytokines, which will induce the differentiation of B cells into fully differentiated antibody secreting cells and memory cells. So let's read that one whole paragraph one more time, okay? I'm just going to leave this picture up and we will read along, basically. So most antigens introduced into the body fall into the category of thymus-dependent antigens. Okay, the response to such thymus-dependent antigen molecules requires the direct contact of the B cells or the TH cells and their cytokines, as you will see. So here's what happens. First thing, there's cross-linking of B cell surface receptors with the antigen. That material is endocytosed and processed via the endocytic exogenous pathway to generate MSC class 2 peptide conjugates, which are then inserted into the membrane, just as we have seen during phagocytosis and processing in the professional phagocytic lineages, such as macrophage, neutrophils, natural killer cells. So similarly, that's the same thing happens here. Simultaneously, expression of co-stimulatory molecules, such as B7, is upregulated on the B lymphocyte, making them effective presenters of the antigen to the Th cells in the area. Once a Th cell recognizes a processed antigenic peptide displayed with the MHC2 class 2 molecules on the membrane of a B cell, the two cells will then form a conjugate, and the Th cell is activated and induced to become a Th2 cell. Now, this Th2 cells in conjugates Th2 cells in conjugates with a B cell will then rearrange their Golgi apparatus towards the junction with the B cell, leading to the directional release of cytokines towards B cell. In addition, expression of a molecule known as CD40L on the surface of Th2 cell is upregulated. And this molecule interacts with the CD40 on the B cell to provide the second signal for B cell activation. B cells are encouraged then to proliferate after this interaction of CD40 and CD40 ligand. The final signal delivered by the Th2 cell is the release of cytokines, which will induce the differentiation of B cells into fully differentiated antibody secreting cells and memory cells and induce class switching. They are differentiated, start producing antibodies, and will switch class. Okay, so we need to know what are the three steps. Let's see in a nutshell. Humoral immunity is basically talking about antibodies. Defense against extracellular agents. B lymphocytes are attracted to and to follicular areas of the secondary lymphoid organs. Pathogens are thymus-dependent antigens. B cells contract with Th cells. B cell contact with Th cells requires that there is a MHC2 with a peptide presentation, that there are co-stimulatory molecules present, B7, that there's CD40 and CD40L binding. Th2 cytokines induce B cell differentiation, memory, and class switching. So it's not telling me what the steps are basically. So the first signal for the activation of the B lymphocyte is the initial cross-link of the idiotypes of B cell with the T cell. So this is the first signal right there. Signal 1. Okay. Second signal, let me find it. Here we go. This is the second signal. CD40 and so this is signal number 2 right there. So this is first signal, signal number 2. And again, B7 and CD28 is necessary so that you don't have energy. You do have a response. That has to bind. I think it's almost like an anchoring or saying, all right, you have my go ahead. This credibility, basically. That's what it's testing for. This is just giving the go ahead. Then basically the T cell will position its Golgi on the side and start producing cytokines. And we know that they produce IL-4, 5, 6, 13, 10, and TGF-beta. They will act on this B cell and induce that B cell to start producing, to first of all getting fully differentiated. Then they will obviously start secreting antibodies and become memory cells, and at the same time cause class switching from IgM, for instance, to IgG, Okay, so now let's move on. T cells recognize peptides bound to MHC molecules. Therefore, antigens that possess no peptide structure cannot be recognized by T cells. These antigens are called thymus-independent antigens. Okay, and induce lipopolysaccharide from the cell envelope of gram-negative bacteria and polysaccharide capsular antigens. Okay. So we know that T cells will only recognize peptides bound to MHC molecules. Agreed. So if there's an antigen that has that possess no peptide structure cannot be recognized by T cells. So if an antigen has no MHC, basically, so they're called thymus independent antigens. Those were dependent, meaning they required an MHC. Now, if they don't have something carrying it, then they are the lipopolysaccharide from cell envelope of gram-negative bacteria, or they could be a polysaccharide capsular antigen. These are the only type of antigens that would be thymus independent, meaning they will not bind. They don't have peptide, and thus they do not bind MHC molecules. These antigens may directly stimulate B cells to cause proliferation and secretion of antibody. Wow. Or they may act as B cell mitogen, directly causing mitosis regardless of the cell antigenic specificity. Yoja. Oh, that's something. The response to thymus independent antigens is generally weaker than the response to other classes of antigens. The response to thymus independent antigens generally weaker than the response to other classes of antigens, resulting in the secretion of IgM antibodies only and the absence of immunologic memory. Okay, so you have thymus dependent and thymus independent. Thymus dependent will cause, the MHC binding, and then MHCs to presents it to the T cell, and then there's a major response. There's memory, there's plasma cells, there's antibodies, a specific antibody. Whereas when you have thymus independent, basically they can be things like lipopolysaccharide or polysaccharide capsular antigens, and um, they can bind B cells and stimulate them to start undergoing mitosis, or even causing further. It doesn't matter what the specificity of that whether or not they're specific even to that antigen. Wow, so idiotype doesn't matter at all. Go figure. And basically, this is not going to be very strong because there's no memory, there's no plasma cells being formed, and obviously the antibodies will always be IgM. So, in a nutshell, thymus independent antigens contain no peptides, stimulate only IgM, and create no memory. Okay. Although all of the antibody molecules secreted by a clone of B lymphocytes will have identical idiotypes, the B cell is induced to make new classes or isotopes of immunoglobulin in response to cytokine directed instruction from the Th2 cell. Okay. The progression of the new antibody isotypes produced by B cells is defined by the sequence of constant domain coding in the B lymphocyte DNA. And each isotype of immunoglobulin is designed with a different effector function in mind. Just as the three-dimensional structure of the idiotype defines the antigenic specificity, the sequence of amino acid in the constant domains of the immunoglobulin molecule or the isotype dictates the effector functions that will be expressed. That was redundant. Okay. So basically, they still have the same idiotypes, but now when you switch classes, you're changing isotypes. So IgM could be turned into, or IgD could be turned into IgG, IgA, IgE, depending on what's needed. Okay, so the biologic function of segments of the antibody molecule was first elucidated by the digestion of these molecules with proteolytic enzymes. Okay, if an antibody molecule is digested with papain, Cleavage occurs above the disulfide bonds that hold the heavy chains together. This generates three separate fragments, two of which are called FAB, fragment antigen binding, and one is called FC, fragment crystallizable. Cleavage of the antibody molecule with pepsin generates one large fragment called FAB, F with two ABs, okay, and a digested FC fragment. Okay, the bridging of antigens by antibody molecules is required for agglutination of particulate antigens or the precipitation of soluble antigens. Okay, so why are we talking about? Is this how we're going to make different isotypes? Probably that's what it seems like. So if you cut the any antibody, IgM or IgD, and you cut it above the disulfide bonds, you end up with two of those FABs, um, two fragments that can bind antigens, and one fragment that is crystallizable. But if you cut that antibody with pepsin, you end up with one large fragment called FAB and another FC fragment. So it's probably below the disulfide bond. Um, okay. Now we're talking about bridging of antigens by antibody molecules is required for agglutination of particulate antigens or the precipitation of soluble antigens. Okay. Mitogens activate many clones of B cells and are used clinically to assess lymphocyte function. In a nutshell, isotype switching is directed by Th2 cells. Yes, isotypes dictate effector function of the antibody molecule. Okay. Oh, here we go. Isn't that nice? There's a nice picture here. See, papain is cutting above so that you end up with 1, 2, 3. That's exactly how I imagined it. And pepsin sites bind underneath and they cut there and you end up, oh, hello. So there are many different sites for that. So you end up with one big FAB and then you have many different fragments of FC. Papain generates two FABs and one FC. FAB is monovalent, capable of binding to antigens. Pepsin generates one FAB with two antigen sites. FAB is divalent and capable of binding and bridging. I see. IG and FAB2 fragments, IgG and FAB2 fragments both have a valence of 2 and can bridge between antigens. Right, I think I better read this part maybe. So we're going to learn how to cut them up, I suppose. I guess we'll probably come back to this. Okay, the primary humoral response. The first isotype of immunoglobulin that can be produced by a B cell with or without T cell help is IgM. Agreed. This is because coding for constant domains of the heavy chain of IgM with mu chains are the first sequences downstream from the coding for the idiotype of the molecule. The IgM molecule on the surface of the B cell is a monomer, but the secreted form of this molecule is a pentamer, held together in an extremely compact form by a J chain synthesized by the cell. So, really, look at that. That's it? One J chain? Okay. So you have a whole bunch of IgMs. We understand how that was made. But in between, you have these binding things, and that the two ends are joined with a J chain. You see J light chain variable constant, mu heavy chain variable constant, one big IgM monomer. What is this line? That is what I'm curious to know. Okay. The design of the IgM pentameter maximizes the effector functions critical to the body early during antigenic challenge. Because of its multimeric structure, which is 5 of the Y-shaped monomers joined into one unit, plasma IgM has 5 times the capacity for binding antigenic epitopes as any monomeric immunoglobulin unit. The valence of the molecule is therefore 10. In other words, 10 identical epitopes can be simultaneously bound, as compared with 2 for the monomeric structure. This makes IgM the most effective immunoglobulin isotype at sponging the free antigen out of the tissues and proves critical as the humoral response evolves in trapping antigen so that it can be presented to the lymphocytes that will ultimately refine the choice of effector mechanisms. That I agree with. Okay, so remember that the valence is just how many idiotypes sites you have available. Although the binding strength, the affinity of the idiotype for the epitope may not be strong early in their immune response, the IgM molecule possesses the highest avidity of any immunoglobulin molecule produced in the body. What does avidity mean? Avidity is the number of combining sites available to bind epitopes. Isn't that the same as the valence? So the valence of the molecule is how many you can hold. Avidity is number of available sites. Okay, so plasma IgM valence is 10, functions in trapping free antigen. Affinity or binding strength may be low. Avidity or multi-point binding, highest of all isotypes. Okay, so low affinity, but definitely more available binding sites. How is this different than from valence? The valence of the molecule is 10. Capacity versus number of sites, isn't that the same thing? I don't know. Okay. Here we have affinity. Affinity, I understand how well you bind, which is low. Avidity is the number of sites. Agreed. But what about avidity and valence? All right, so the multimeric structure of IgM also makes it the most effective antibody at activating a complement, a set of serum proteases important in mediating inflammation and antigen removal. So it's very good at activating complement because it's multimeric structure. And complement are basically a set of serum proteases important in mediating inflammation and antigen removal. So complement is basically a protease. Okay. Serum IgM is incapable of binding to cellular Fc receptors. Right, because they're all on the inside, right? And thus cannot act as opsonin, or a mediator of antibody-dependent cell-mediated cytotoxicity. So in a nutshell, IgM is most effective isotype at activating complement. It is not an opsonin. It does not mediate antibody dependent cell mediated cytotoxicity. Okay, that seemed pretty. The only thing I want to know is the difference between valence and avidity. And what in the previous section, I wanted to know what are these effector functions they keep talking about. Okay, isotype switching. As the B lymphocytes receives cytokine signals from the activated Th2 cells in the secondary lymphoid organs, it is induced to undergo isotype switching, changing the heavy chain constant domains to classes of antibodies with new and different effector functions. It does this by rearranging the DNA encoding the constant region of the heavy chain by activating switch regions that cause the intervening DNA to be looped out, excised, and degraded. So it's at the DNA level. That's where you rearrange it again. So that cells, once it's rearranged, can't go back, basically. The idiotype is then joined to a new constant region domain coding and an antibody molecule with identical antigenic specificity, but a new effector function is produced. This isotype switch is one way. Because the excised DNA is degraded, a cell that has begun to produce an isotype downstream from IgM coding can never produce IgM again. Got it. Dramatic enough. All right. So basically, here you have your chain, your V variable, diverse joining chain variable domain. You have the mu, the delta, the gamma, the delta, alpha. Anyway, so you excise whatever you want. So here we're excising all of them except gamma 1. So you excise all of this area. So these are called switch regions, huh? By activating switch regions that cause the intervening DNA to be looped out. Activate switch regions. These things are called switch regions. Though I would think it would only be here and here. Why this one? I don't get it, but okay. All right, clinical correlate. IgM is used as a measure of a primary response, acute infection. Convalescent serum will have mostly IgG with subthreshold levels of IgM. Note, isotype switching is induced by Th2 cells. Now, what I want to know is what kind of interleukins, what cytokines are causing what? What cytokines are causing or the selection or switching to a certain isotype, you know, of antibody. Okay. So this is why IgM is the principal immunoglobulin of the primary immune response when antigen is first encountered, and it is replaced in later responses by antibodies of different isotypes. Although IgM antibodies are occasionally produced at low levels during secondary and later immunologic responses, they are always produced by cells seeing that antigen for the first time, namely naive cells newly emerging from the bone marrow. So, although you might have different types of cells, but if you even have another infection with something, if it's producing IgM, it's that cell's first time seeing that antigen always. So that means that you probably have a lower reserve of cells that have not seen that antigen before. So that would mean that there must be some mechanism of saving those, who knows? Okay, so let's read into that. So here we have a diagram which shows the primary and secondary antibody responses. Primary antigenic channel, primary response, secondary response. So first thing, you produce a lot of IgM. Eventually, that starts to die down, and you start generating IgG. That dies down. Suddenly, you have a secondary response, and immediately you'll have a slight IgM, but the secondary response with IgM would be much higher. That is the primary one. Let me read this part again. Maybe I didn't get this. So, IgM is the principal immunoglobulin of the primary immune response when the antigen is first encountered, and it is replaced in later responses by antibodies of different isotopes. Agreed. Although IgM antibodies are occasionally produced at low levels during secondary and later immunologic responses, they are always produced by cells seeing that antigen for the first time. So I'm thinking that means that it's their first time seeing that antigen, that they never had a primary response, those cells in particular. They're always produced by cells seeing that antigen for the first time, namely naive cells newly emerging from the bone marrow. I see. So new cells see a new an antigen that they think is, wow, this is a new antigen. Look at what I found. But guess what? It's already been discovered before by a B cell before, and that one's already mounting an immune response. I wonder if those old cells are killed off, or maybe they'll bind stronger and they'll be kept. Who knows? Anyway, during the activation of B lymphocytes by Th2 cells, intense proliferation of the B cells results in the formation of germinal centers in the follicles of the lymph nodes and spleen. These are clones of proliferating antigen specific cells. During the intense proliferative response of the B cell, random mutations in the coding of the variable domain region may occur. Is that good or bad? This is called somatic hypermutation and creates single point mutations in the antibody idiotype. Well, on the one hand, it might make it stronger, but on the other hand, it might make it worse or even deadly, you know? So somatic hypermutation is what happens when you have a lot of B cells rapidly proliferating in their germinal centers. If these slightly altered idiotypes have an increased affinity for the antigen, then the cell expressing them will be at a selective advantage in competing to bind the antigen naturally. Because binding antigen serves as the first signal for proliferation, over time, clones of cells with higher receptor affinity will begin to predominate in the germinal center. So this clonal selection results in the predominance of clones capable of producing antibodies with increasing affinity for the antigen, a process known as affinity maturation. Okay, this means that although isotype switching will necessarily decrease the avidity of the preponderance of antibody molecules as the immune response evolves, this will be substituted by an increase in antibody affinity over time. Okay. All right, so basically, Th2 cell tells the B cell, all right, time for you to multiply. It starts to multiply and forms the germinal centers. But while it's multiplying, it might have mutations in its DNA in the variable domain region, and this would be called somatic hypermutation. And over time, if that mutation is very, very good or has a high affinity for that particular antigen, eventually that will start to dominate over time compared to the others. And this will be clonal selection for that specific clone of B cells. And they will start producing antibodies with a higher affinity for that antigen. And this is called affinity maturation. So, although you have a less amount of avidity, there is an increased amount of affinity. So, it's more specialized. Okay, so in a nutshell, germinal centers are clones of proliferating antigen specific B cells. Somatic hypermutation may cause minor idiotype changes. Clonal selection by competition for antigen causes affinity maturation. Got it. The choice of activation of particular switch regions in the B cell DNA sequence is apparently dictated by the release of specific cytokines by activating activated Th2 cells. Okay. The choice of activation of particular switch region in the B cell DNA sequence is apparently dictated by the release of specific cytokines by activated Th2 cells. Yeah, I guess that sounds right. The Th2 cytokines dictate switch region activation. Yeah, but which ones, right? Okay, come on. Okay. Here we see a picture Th2 cell direction of B lymphocyte proliferation and differentiation. So you have an activated B cell, centroblast, and they're told to proliferate by Th cells. And they differentiate and they continue to produce IL-5, 4, and 13. Oh no, they're stimulated by 4, 5, 13 TGF beta. And if it's a Th5, I mean IL-5, it'll make it into IgA. That's what I'm talking about. IL-4 and 13 together will make it into an IgE. And if it's nothing, then it will be IgG. And what is this? I don't know what this is. It's just like a stem cell of some kind. I don't know. 5 always plus forever. For always and forever. Huh, that's cute. Memory tools. For always, 5 always and forever. Cute. IL-5 will cause the formation of IgA. IL-4 will cause the formation of IgE. But what about IL-13? That also does IgE. But I want to know what this is about. What's this cell doing? Okay. Clinical correlate. X-linked hyper IgM syndrome is characterized by a deficiency of IgG, IgA, and IgE. Okay. And elevated levels of IgM. IgM levels can reach 2,000 milligrams per deciliter. Normal is 45 to 250 milligrams per deciliter. It is most commonly inherited as an X-linked recessive disorder, but some forms seem to be acquired and can be seen in both sexes. Okay, because X-linked recessive would be seen in mostly guys, right? Males, children. The peripheral blood of infected individuals has high numbers of IgM secreting plasma cells, as well as autoantibodies to neutrophils, platelets, and red blood cells. Okay, these patients fail to make germinal centers during a humoral immune response. Children with this condition suffer recurrent respiratory infections, especially those caused by pneumocystis jirovecii. The defect in this syndrome is in the gene encoding the CD40 ligand, which maps to the X chromosome. Therefore, Th cells from these patients will fail to express functional CD40L on their membrane and will thereby fail to give the co-stimulatory signal necessary for the B cell response to T-dependent antigens. So only IgM antibodies are produced. The B cell response to T-independent antigens is unaffected. Okay, I need to read this again. X-linked hyper IgM syndrome is basically you have a deficiency of IgG, IgA, and IgE. You see, I don't see like okay, I understand 5 and TGF beta making IgA. 4 and 13 making IgE and nothing was making IgE, but then I don't know what this is. Okay. All right, so IgM levels are really, really high, and all other Ig antibodies are really, really low. It's an X-linked recessive disorder, but can be seen in both sexes. The peripheral blood of infected individuals has high number of IgM secreting plasma cells, that's understood, as well as autoantibodies. They have autoantibodies to neutrophils, platelet, and red blood cells. Well, that's not good. These patients fail to make germinal centers during humoral immune response. Children with this condition suffer recurrent respiratory infection, especially those caused by Pneumocystis jirovecii. The defect in this syndrome is in the gene encoding the CD40 ligand, which is on the X chromosome. Really? Therefore, the Th cells from these patients will fail to express CD40L on their membrane. So it's a problem with the T cells and will thereby fail to give the co-stimulatory signal necessary for the B cell response to the T-dependent antigens. So only IgM antibodies are produced. The B cell response to the T-independent antigens is unaffected. So the X chromosome has a defective CD40 ligand because of which it can't bind the B cell CD40. And because it can't bind, it can't start producing, rearranges Golgi and starts secreting interleukins towards B cells, telling it to form either a plasma cell or a differentiate to start forming, switching its class and start producing IgG, IgA, or IgE antibodies. So the B cells just continue to produce IgM constantly till they become like 2000. But that does not explain why you have autoantibodies to neutrophils, platelets, and red blood cells. Don't know about that. Okay, good to know. One thing I want to do, because there was this one part that I kind of skipped over. IgG and FAB2 fragments both have a valence of 2 and can bridge between antigens. Soluble antigens will then precipitate. Particulate antigen, red blood cells, and latex beads will then clump together or agglutine. Did I miss something? I don't know. Seems like maybe I missed something. No, I did not. The bridging of antigens by antibody molecules is required for agglutination of particulate antigens or precipitation of soluble antigens. And then you go on to say that soluble antigens precipitate, particulate antigens clump together or agglutinate. So precipitate is just like fall out. And particulate antigens, red blood cells and latex beads, they agglutinate. And IgG and FAB2 fragments both have a valence of 2 and can bridge between antigens. Okay, gotcha. All right. And this was important because he had somatic hypermutation, which is a mutation that occurs while the B cell is crazily reproducing itself. Clonal selection is when one antigen binds to a particular antibody with a higher affinity, so that over time, that particular B cell producing that particular antibody is selected for and ends up becoming in higher number over time. Um, Affinity maturation is basically increasing, producing antibodies with a higher affinity every time. Okay, and here, oh, and we're done. Next subject, antibodies of immune responses. The preponderant isotype of immunoglobulin that begins to be produced after IgM during the primary immune response is IgG. The preponderant isotype, so we're talking about antibodies of secondary immune responses. Okay. The preponderant isotype of immunoglobulin that begins to be produced after IgM during the primary immune response is IgG. IgG is a monomeric molecule with a gamma heavy chain and a new set of effector functions. The preponderant isotype of immunoglobulin that begins to be produced after IgM during primary immune response is IgG. Duh. IgG is a monomeric molecule, yes, with a gamma heavy chain, yes, and a new set of effector functions, obviously. IgG exists in 4 different subisotypes or subclasses in humans. You have IG1, IgG1, IgG2, IgG3, and IgG4, each of which exhibits slightly different capacity in effector function. But in general, IgG activates the complement, acts as an opsonin, and mediates ADCC, which means antibody-dependent cell-mediated cytotoxicity. All right. It is also actively transported across the placenta by receptor-mediated transport and thus plays a crucial role in protection of the fetus during gestation. IgG is the major antibody produced after IgM. IgG exists in 4 subisotypes. IgG activates complement, opsonizes, and mediates antibody-mediated, antibody-dependent cell cytotoxicity. IgG is actively transported across the placenta. Here's the basic structure of the IgG. We see here you have a heavy chain and a light chain. Heavy chain has a constant region and a variable region, and the light chain has a constant region and a variable region. These are the idiotype where the antigen can bind. This is the hinge region. This is the double bond. This can be kappa or gamma. This will definitely be the gamma, constant gamma heavy chain. This will bind the complement binding region here, cell receptor binding region here. So binds complement here, cell receptor here, hinge region, constant heavy chain domain, variable heavy chain domain, light chain, constant light chain. Okay. Another isotype of antibody that can be produced following class switching is IgA. Although this isotype is much more commonly produced in the submucosa than in the lymph nodes and spleen. IgA generally exists as a dimer held together by a J chain similar to that produced with IgM and serves as a major protective defense of the mucosal surfaces of the body. Its sole function appears to be the inhibition of binding of toxin or adhesive microbial components to the mucosa of the digestive, respiratory, and urogenital system, and it does not activate complement or act as an opsonym. So IgM and IgG do act as a complement. IgG does activate complement, and it is also an opsonym. Okay, so here we have an IgA dimer. Looks kind of like your usual antibody, but there are two of them fused together. And they're fused on their, well, look at that. I don't know, maybe these are both beta or maybe they're alpha and beta. Can't tell. In a nutshell, most IgA is produced in the submucosa. IL-5 and TGF-beta cause isotype switching to IgA. IgA is a dimer with a J chain. IgA inhibits binding of adhesive substances to mucosal surfaces. IgA is an important component in breast milk. See here, they say IL-5 and TGF-beta. What did they say? They did not say anything about IgG, did they? Nothing. Nothing is triggering that production. Okay. Moving on. The homing of specific memory cells to epithelial and mucosal surfaces leads to the production of specialized lymphoid aggregations along these barriers, collectively referred to as mucosal associated lymphoid tissues. So here basically you have memory cells that tend to home or hang out near the epithelial and mucosal cell, which makes sense because that's where most of the antigens will be coming in from. Um, so the MELT, mucosal-associated lymphoid tissues, include the tonsils, the and Peyer's patches, as well as numerous less well-organized lymphoid accumulations in the lamina propria. The Th2 cells in these sites are dedicated to providing help for class switching to IgA. So these Th2 cells will be constantly producing IL-5 and TGF beta. Um, most IgA secreting, most IgA secreting B lymphocytes and plasma cells in the body will be found in these locations. Secretory IgA, that which is released across the mucosa of the respiratory, digestive, and urogenital tracts, differs from the serum IgA in an important fashion. As the IgA dimer is produced by plasma cells and B lymphocytes, it becomes bound to receptors on the abluminal side of the epithelia. What is the abluminal side of the epithelia? Probably the one facing the lumen. It is then endocytosed and is released into the lumen wearing a secretory piece that is a residue of the epithelial cell receptor. Oh, yay, there's a picture. Thank God. All right, so let's do this again. So the B cell is producing the plasma cells and the B cells, right? They're producing this IgA dimer. Well, it will go ahead and get attached to the receptors on the abluminal side of the epithelia. It will then be endocytosed by the epithelia and go through the epithelial cells and be released exocytosed into the lumen, wearing a secretory piece that is the residue of the epithelial cell receptor. So it wears a little hat, I suppose. Interesting. The secretory component thus serves an important function in transepithelial transport, and once in the lumen of the tract, has a function in protecting the molecule from proteolytic cleavage. So, so most of these memory cells will home towards the epithelial and the mucosal surfaces, and they will form specialized lymphoid aggregations like mucosal associated lymphoid organs, such as tonsils, Peyer's patches, and other things in the lamina propria. Th2 cells will also be there, and they will just help whenever they can so that these cells, memory cells, can start producing IgA. Here you will have, in these locations, that's where you find most of the B cells and most of the plasma cells. Secretory cells, um, they are basically produced by plasma cells and B lymphocytes. They differ from the serum IgA in an important fashion. What do you mean serum IgA? I'm thinking if they're secreted, where would they be secreted if not blood? You know? Okay. So anyway, they come to the abluminal side of the epithelia. They get endocytosed by the epithelia. The epithelial cell then put on this little hat, well the receptor that they bound to in the initial place, and they keep that on forever. Once in the lumen of the tract, this has a function of protecting the molecule from proteolytic cleavage. Okay. So malt contains Th2 cells assisting in IgA production. Secretory IgA is a dimer with secretory component. Secretory component is basically transepithelial transport protection from proteolytic cleavage. Understood. So you have the plasma cell producing IgA. I'm thinking it's going, this is the mucosa, they're saying. Then this is the epithelial cells under, obviously, the mucosa. So they're entering tissues, I think. IgE is a so-called homocytotrophic antibody. Homocytotrophic antibody because it binds directly to the Fc epsilon receptors present on the mast cells and basophils without binding antigens. Okay. So they don't need to bind an antigen, they just directly bind to the cells, the basophils and the mast cells, and is involved in the elicitation of protective immune responses against helminth parasites and many allergic responses. It does not activate, complement, or act as an opsonin. Its heavy chain is called an epsilon chain. Okay. IL-4 and IL-13 cause isotype switching to IgE. IgE is bound to mast cells and basophils. IgE mediates immediate type 1 allergic reaction, and IgE protects against parasites. Oh, wow. Here we go. Summary of the biological functions of antibody isotypes. Okay. So here we have all the antibodies, M, G, A, D, and E. I don't know why they chose this order. But what is the heavy chain? Basically, that's understandable. This is mu, gamma, alpha, delta, epsilon. Adult serum levels in milligrams per deciliters. So the most trace amounts of D and E usually in serum, but the most would be IgG always. Second would be IgA, and third would be IgM. So what are their functions? Complement activation of the classic pathway. Yeah, M and G would both do that. Nobody else will do that. Opsonization. Everything, no, nothing opsonizes except IgG. Antibody dependent cell mediated cytotoxicity. Again, only IgG. Placental transport, IgG only. Naive B cell antigen receptor. That's M and D, right? Memory B cell antigen receptor. One only? What do you mean one only? Memory B cell antigen receptors. Yeah, so yeah, that would be GANE, right? Yeah. Trigger mast cell granule release. Only E, right? Yeah. Okay. Immunodeficiency involving B lymphocytes. Patients with B cell deficiencies usually present with recurrent pyogenic infections. Patients with B cell deficiencies usually present with recurrent pyogenic infections with extracellular pathogens. The absence of immunoglobulins for opsonization and complement activation is a major problem. So, the only immunoglobulins that opsonize is IgG, and the only things that activate complement is IgM and IgG. Okay, so the T cell immune system is intact, and T cell activates activities against intracellular pathogens. Delayed type hypersensitivity and tumor rejection are normal. So we're just talking about B cell deficiencies. They have recurrent pyogenic infections with extracellular pathogens. And because they don't have B cells, um, you don't have opsonization, you don't have complement activation, and instead you have the T cell available, and that can still activate against intracellular pathogens. They can cause delay type hypersensitivity, and they can still cause tumor rejection. But you cannot opsonize, you cannot activate complement, and you just end up with recurrent pyogenic infections. Complement. The complement system is a set of interacting proteins released into the blood after production in the liver. So the complement is produced in the liver. The components act together as zymogens, meaning that they activate one another in cascade fashion after initiation from a variety of stimuli. The two different pathways of activation occur in the body and culminate similarly in the production of important split products that mediate inflammation, enhance phagocytosis by opsonization, and cause lysis of particles by membrane pore formation. So the complement system has two pathways of activation. It enhances inflammation and phagocytosis, and in the end, its job is to cause lysis of that cell by forming a pore. All right. So here we have the two pathways. One is the alternative, and the other is classical, right? They make it sound so easy. I remember I used to struggle with this so much. I don't want to speak before. I don't want to speak to Sue meaning. All right. So alternative pathway of complement activation. Okay, so I should probably look at this picture first. Recruitment of inflammatory cell. We know C5a obviously from before, but look at this. You also have C3a and C4a. So 3, 4, and 5a end up recruiting all inflammatory cells, right? Opsonization of pathogens would be by C3b bacteria, clearance of immune complexes. What? Okay, so C3b will surround a bacteria, or they will bind an antigen, which will then bind uh, immunoglobulin and form an immune complex. 3. Killing of pathogens, C5b. C3b was opsonization, C5b is membrane attack complex, puts holes in the membranes. Okay. The alternative pathway of complement activation is probably the more primitive of the two pathways because it is initiated by simple attraction of the early factors to the surface of microbes. Are we talking about C3b? The bacterial polysaccharides and the lipopolysaccharides of the cell envelope of gram-negative bacteria both serve as potent initiating stimuli. Okay. So if you have a lipopolysaccharide or polysaccharide, Coming from a bacteria, it's going to stimulate the alternative pathway. Uh, so the alternative pathway is initiated by surfaces of pathogens. So C3A, C4A, and C5A are all anaphylatoxins. While only C5A is also a chemotactic. Yep, but you said they all were chemotactic. Recruitment of inflammatory cells. C5A is chemotactic. Sounds like they all are. I don't know. Okay. So here we have a picture. Here is, whoa, okay, where to begin? Let's see. You have here a spontaneously generated C3b. Okay. Microbial surface, example, endotoxin protects C3b. Cell membrane of bacterium. So this is the cell membrane of the bacterium. This is the microbial surface. Okay. So you suddenly have this C3b just floating around, and it will join with this factor B. And they will join together. And in the presence of factor D, the B's little a, the small part of that B, will come out. And they will form 3B and BB. C3 convert. I love this diagram. I've never seen it presented in this way. Wow, no wonder Kaplan's awesome. Okay, this is very cute. Okay. So you have a C3, right? And C3 has a C3A and it has a C3B. So C3A, so you have a C3 and a factor B, right? So C3B will join with the whole factor B. And then in the presence of factor D, the BA part will come off. And in the presence of C3 convertase, either way, they can be separated back into a C3b. Amplification loop, it's called. All right, similarly, this will continue on in the presence of something, maybe BA, maybe? And that will cause the joining of this new thing here, 3B, BB, and another 3B. Okay, C5 converted, C3b deposited on cell particle surface. Eventually you become C5b and 5. This is not making much sense to me. I'm sorry. I tried. Okay. Moving on. The classical pathway is activated by antigen-antibody complexes and is probably the more phylogenetically advanced system of activation. Both IgG and IgM can activate the system by this pathway, although IgM is the most efficient. Okay. Classical pathway is activated by antigen-antibody complexes and is probably the more phylogenetically advanced system of activation. Both IgG and IgM can activate the system by this pathway, although IgM is more efficient. Is it? Interesting. All right. So here we have the classical complement pathway. I hope I understand this better. So you have complement components in blood, such as C4, C1, C2, even C3 there. You have C4, C5, and C6 through 9. Okay. So first up, IgM or greater than or equal to 2 IgGs have to be there. And basically C1 will bind it, and then C4 will act on it. Ca comes off, C4b stays on. Next C2 joins it, and C2b comes off, and 2a remains. Next you have C3. Its little tiny part leaves, which would be C3a comes off, C3b stays, C5 then joins, and suddenly you have more complements joining. C3 convertase, C5 convertase. Oh, you need these convertases in the process. Anyway, B fragments are usually membrane surface bound. Okay. Excess C3b deposited on cell particle surface binds C3b receptors on phagocytes. Membrane attack complex puts holes in membrane. Okay. Although the complement cascade is considered a component of the innate immune response, its overlapping stimulation of effector functions of cells of the adaptive immune response, as well as its role in enhancement of inflammation, make it a critical effector system for removal of extracellular invaders and concentration of antigens into the secondary lymphoid organs, where the adaptive immune responses are elicited. When uncontrolled activation of complement occurs in certain disease states, damage to host tissues can ensue. Physiologic controls on complement activation occur at the level of C1 classical pathway and C3 and C5 both pathways. That's a big sentence. Although the complement cascade is considered a component of the innate immune response, its overlapping stimulation of effector functions of cells of the adaptive immune response, as well as its role in the enhancement of inflammation, make it a critical effector system for removal of extracellular invaders and concentration of antigens into the secondary lymphoid organs, where the adaptive immune responses are elicited. So they're good in between. When uncontrolled activation of complement occurs in certain disease stages, damage to host tissues can ensue. Physiologic controls on complement activation occur at the level of C1, classical pathways, and C3 and 5, which is found in both pathways. So here we go, talking about complement deficiencies. Genetic deficiencies have been described for each of the components of complement and their regulatory proteins. These deficiencies highlight the critical role of the early components of complement in generating C3b and the essential role of C3b for the clearance of immune complexes from the body. Furthermore, even though gram-positive bacteria may be resistant to the membrane attack complex of the complement, the early components of the cascade mediate localized inflammation and opsonize the bacteria. In a great number of cases, then, it is the complement cascade that converts immunoglobulins into powerful effectors of bacterial destruction. So the complement is important for localized inflammation, It's important for opsonizing bacteria, um, and whatever complement cascade might follow. You have the conversion of immunoglobulins into powerful effectors of bacterial destructions. When deficiencies of complement regulatory components occur, then the uncontrolled activation of the complement cascade can have dangerous results in the body's own innocent bystanders. Oh no. In the hereditary angioedema, uncontrolled complement activation at the mucosal surfaces causes edema and pain. Okay. So there's something called hereditary angioedema, and here you have uncontrolled complement activation at mucosal surfaces. And as a result, you have a lot of edema and pain because of the ensuing inflammation. In paroxysmal nocturnal hemoglobinuria, the absence of regulatory proteins causes paroxysms of hemolysis of red blood cells and the resulting hemoglobulin urea. Okay, gotcha. Here we have, what is this? This looks like overview of the generation of humoral effector mechanism. I hope it's very detailed. Let's see. Here we have an antigen, lottie dottie dog, coming on in. It meets with an antigen presenting cell, is displayed on MHC, binds to CD4. Well, obviously, this is a B cell because it's a CD40 coming out of it. And then that THC cell, TH1 does nothing. This will become into a TH2, not TH1, TH2 cell. So immediately, what we have is the binding of CD40 ligand with CD40, and thus the release of all these cytokines, which will act on that B cells. This B cell will become activated and start producing all these other B cells, and then eventually they will become plasma cells, which will start producing a specific um, antibody. Um, and now this antibody will join with the complement, which would be C3, right? There it is. C1. Sorry, C1. It will bind with C1 complement, which will then activate the classical pathway and form the MAC lysis. It can also bind with C3b and cause phagocytosis. It can also induce, go under, wait a second. Apparently, it's also able to go into alternative pathway, huh? Okay. Um, and it can also go ahead and bind to macrophages where they will be ingested. Gotcha. Oh, we're done? Okay. Humoral immunity is mediated by antibodies synthesized by B cells and secreted by plasma cells. Humoral immunity is the major defense mechanism against extracellular microbes and toxins. Most naturally occurring antigens are thymus dependent, meaning they require collaboration of T helper cells and B cells. Contact between specific B and Th cells involve MHC class II peptide presentation, co-stimulatory molecules B7 and CD28, CD40 and CD40L binding, and cytokine production 2, 4, 5, and 6. Look, it's producing IL-2. Th2 cells direct isotype switching by B cells, which changes the effector function of the antibody produced. Okay. Depending on what they're going to be doing. Thymus independent antigens such as bacterial lipopolysaccharides cross link with the receptors of B lymphocytes and cause them to proliferate and secrete the IgM antibodies. These antigens do not create immunologic memory. IgM is the first isotype of antibody that can be produced. It exists in the serum as a pentamer held together by a joining J chain. The functions of IgM are as a monomer receptor on this B cell antigen capture in the secondary lymphoid organ, and as a pentamer in plasma activation of complement. Th2 activation of B lymphocyte causes intense proliferation in the germinal centers, and the somatic hypermutation may cause slight variation in the shape of the idiotype. Clonal selection of the idiotype with the highest affinity for antigen results in affinity maturation, a general improvement in the goodness of fit for the antigen as the immune response progresses. IgG is the major isotype produced after IgM. It exists in four subisotypes. It activates complement, opsonizes mediated antigen dependent cell cytotoxicity, and is actively transported across the placenta. IgA is the major isotype produced in the submucosa, colostrum, and breast milk. It is a dimer with the J chain holding it together. It functions in inhibiting the binding of substances to cells or mucosal surfaces. It does not activate complement or mediate opsonization. Secretory IgA is transported into the lumen of the gastrointestinal, respiratory, or genitourinary tracts by binding to a polyimmunoglobulin receptor. This receptor, now called a secretory component, is retained for protection of IgA from proteolytic cleavage. IgE is the antibody that binds to mast cells and is responsible for anti-helminthic and allergic responses. Complement is a set of interacting serum proteins that enhance inflammation C3a, C4a, C5a, and opsonization C3b, and cause lysis of particulate substances C5b through 9. The alternative pathway of complement is activated by interaction with microbial surfaces. The classical pathway is activated by antigen antibody complexes. Inappropriate activation of the complement cascade is controlled at the level of C1, C3, and C5. Mhm. Okay. Question: how many are there? I am so ready to, 8 questions. Okay, that's why it's so long. An antibody preparation is being used in a laboratory protocol to study B lymphocytes. The preparation does not activate the cells or cause capping. Okay. It does not cause precipitation of its purified ligand, and it does not cause agglutination of latex beads covalently coupled to its ligand. Which of the following is the most likely antibody preparation? Okay, so you're making antibody, and this antibody does not activate cells or cause capping. I'm thinking you're talking about IgA, who knows? Does not cause precipitation of its purified ligand. Does not cause agglutination of latex, so that's not IgM. Covalently coupled to its ligand. Which of the following is most likely antibody preparation? You know what? I want to do this chapter again tomorrow and see if I do it, because I just don't want to read the answers and then be like, I know. You know what I mean? So I'm just going to end it here. Sounds good.